Hello students, today we will start the lecture second of enzymes and our topic is chemical and biological catalysis. Catalysis is a process which is done by catalysts. Catalysts are chemical substances which alter the rate of reaction and the process is known as chemical catalysis. The catalysis is generally used for the rate enhancement in presence of foreign substances, which itself does not change during the course of reaction and can be recovered as the completion of reaction, at the completion of reaction. In certain cases, the reaction product itself acts as a catalyst. For example, in the oxidation of oxalic acid by acidified potassium permanganate, the velocity increases as the reaction progresses. Here, the term autocatalysis is used because manganese ions are formed during the reaction and which accelerate the rate of the reaction. The exact mechanism by which catalyst enhances the rate of reaction is not understood, but we can see here that the catalyst, what does the catalyst do? Here it is the activation energy for the reaction to occur. But the energy, activation required energy for the reaction to be done, that is the energy required for product reactants to convert into the products is this is the original energy. Now what do catalysts do? The catalyst, in the presence of catalyst, it minimizes the required energy for the reaction to be done, or the required energy for the reactants to change into the products is getting minimized by using catalyst. So catalyst has the effect of it provides that new alternate low energy pathway for the reaction by lowering energy of transition state. Now by the addition of catalyst, the activation energy for the reverse reaction is lowered by exactly the same amount as the forward reaction. So, this explains why catalyst speeds up the both forward and backward reactions to the same extent. If a catalyst doubles the rate of forward reaction, the rate of backward reaction will also be doubled. Hence, the catalyst does not alter the equilibrium, it only hastens, that is, it increases the attainment of equilibrium by speeding up both rates of backward as well as forward reactions to the same extent. Now, what are the characteristics of catalysis? The first, it remains unchanged in the regard to amount the composition at the end of the reaction. Only a small amount of catalyst is required and for example 1 mole of colloidal platinum can catalyze decomposition of 10 to power 8 liters of hydrogen peroxide. Rate of heterogeneously catalyzed reactions increases with the surface area of catalyst. The solid catalyst is more effective in finely divided state. The catalyst does not alter the position of equilibrium in reversible reaction. Catalyst does not initiate the reaction, it only alters the reaction rate. The catalysts are specific for a specific particular reaction. For example, the KClO3 in the presence of manganese dioxide forms KCl and O2, whereas the glucose in the presence of gymase enzyme changes it in the ethyl. Catalysts only alter rates of reactions, they do not initiate them. They only alter the rate of reaction. For example, in the combination of carbon monoxide with hydrogen forming formaldehyde, in the combination of carbon monoxide with the two molecules of hydrogen gas, in the presence of zinc oxide and chromic oxide, it forms the methanol, and the carbon monoxide in the presence of three hydrogen molecules in the presence of nickel form methane. Catalysts work most effectively at certain specific temperature and this is known as the optimum temperature for its working or effectiveness. Certain substances have the effectiveness of catalyst. They are known as catalytic poisons. Catalytic poison are what? They are the chemical substances which decrease the rate of reactions. Now, the type of catalysis. Type of catalysis. Catalysis are of two types, homogeneous and heterogeneous catalysis. And homogeneous, when the both reactants and catalysts are in a single phase. Heterogeneous catalyst, when catalysis, when reactants form one phase and catalyst have the another phase. Usually catalysts are used in the solid phase. Third type of catalysis is the intramolecular catalysis, that is the catalysis done within the molecule. And fourth type of catalysis is multifunctional catalysis. Now we come on the homogeneous catalysis. The very good example of homogeneous catalysis is the hydrolysis of ester. In this catalysis, it belongs to one phase only, that is the surface or the phase boundary is absent. So in the homogeneous catalysis, we see here the ester in the presence of acid combines with water molecule and form acetic acid. So what does the proton do here? The proton reacts with the ketonic, the ester form and form a enolate ion. Here, in the benzoin condensation, when the benzaldehyde reacts with the cyanide ion, it forms the benzoin. The benzoin condensation involves following which cyanide ion attacks on the aldehydic carbonyl carbon atom, and this carbonyl atom then reacts then get peroxide ion, and then the whole mechanism is benzaldehyde cannot attack directly a second molecule on the same as the hydrogen in benzaldehyde is not. We can see here the whole mechanism of the benzoin. Homogeneous catalysis may be specific acid, specific base, general acid or general base catalysis. Here are the mechanism for the specific. This is the specific acid catalysis, and this is the specific base catalysis, and it is the general acid base both catalysis. Now we come on the heterogeneous catalysis. When substrate and catalyst are different phases, catalytic process is known as heterogeneous catalysis. For example, hydrogenation of ethylene in presence of metal catalyst such as palladium, platinum or nickel. In heterogeneous catalyst, role of catalyst is to bring together ethylene and hydrogen molecules in order to make the reaction feasible. Here, the reactants have affinity for catalyst, therefore they get adsorbed on the surface of catalyst. But products do not have affinity for catalyst, hence they get desorbed and released. Heterogeneous catalysis are important in the synthesis of polymers, plastics, as well as in the plastic industry. Ziegler-Natta catalyst for polymerization of olefins is an important example of heterogeneous catalyst. And also, this catalyst is formed by the coupling of TiCl4 and titanium 4 is the coordinating transition metal, and organometallic moiety act as activator by alkylating active sites, that is titanium ion, and removing one chlorine atom. And now the third type of catalysis is intramolecular catalysis. When catalyst as well as substrate are in the same molecule, catalysis is known as intramolecular catalysis. For example, amino group participate in the hydrolysis of ester, and four types of intramolecular catalysis mechanisms are feasible. Here, this is the amino ester, and this amino group attacks on the ester and gives the reaction. Intramolecular general base catalysis is the what that amino group attacks on the proton of the water molecule, and this hydroxide ion will act as a catalyst and attack on the ester group. In the intramolecular general acid specific base catalysis, what happens? The N positive ion which is attached to the carbonyl group of the ester, and it is attacked by the basic hydroxyl group, Then this carbonyl carbon gives the electron to the positive nitrogen. The electrostatic facilitation also occurs here, and here this carbonyl oxygen gives the electrons or support to the positive nitrogen, which is quaternary, which is becoming quaternary here. So intramolecular acid-base catalysis catalyze the reactions in organic systems, reactions are of second order, whereas formation of enzyme substrate complex is a first order. In the furthermore, cyclization relieves large ground state strain. Moreover, effective concentration of carboxyl group in the proximity to hydroxyl group is much more in comparison B to A, which is responsible for more effectiveness of intramolecular catalysis in enzymatic. Here we look that what is happening in the reaction A. In this reaction, what is happening? In B type of reaction, what is happening here? The intramolecular, these are the intermolecular reactions, and this is the intramolecular reaction. The carboxylic group within the molecule reacts with the OH group of the molecule. The steric compression is favored more. Now, multifunctional catalysis. This catalysis is based upon the fact that the several functional groups in the active sites are, if properly oriented with the substrate, concerted catalysis may take place. Concerted catalysis may take place here. Here is a classic example of multifunctional catalysis. It is the hydrolysis of monosuccinate ester of hexachlorophene. This is the multifunctional catalysis. Here the carboxylate ion, which is attacking here in the on the next carboxylate group, and here the catalysis is being done. Bifunctional catalytic mechanism have been studied in detail by Lee and Schmier on acyclic immediate ester. Hydrolysis proceeds through the formation of tetrahedral intermediates, which is redetermining step and subsequently breaks down to an amide or ester. Now, the theories of catalysis. There are two theories given for the action of catalyst. First, the active site theory, and the second theory is the intermediate formation theory. What are the active sites? According to this theory, there are active sites on the surface of catalyst which form the unstable bonds with the reactants, the strain in the bonds of reactants. Thus, old bonds start to break and new bonds start to form. When all the new bonds are completely formed, all old bonds break. So, after the completion of reaction, products are released from the surface of catalyst. And this theory explains the catalysis by solids only, because catalysts come from reactive in powder form because of increase in the number of active sites. Here, these are the active sites which upon conversion to powder form, these active sites are become enhanced. In here, 8 active sites are only, and here 16 active sites are present. Second type of theory of catalysis is the intermediate formation theory. That is, it assumes that the intermediate is formed. This theory states the reactants and catalysts combine to form a reactive intermediate, which being unstable due to high energy and breaks down to yield products and setting from the free catalyst for further catalysis. Here the carbon plus sulfur, which are adding here and forming a carbon sulfide intermediate, and which then decomposes into the product. So this is the equilibrium. Here is the equilibrium in all the two stages. So C stands for catalyst, S is substrate, and P is the product, and C as a star is the reactive intermediate. Now we come to biological catalysis. Enzymes are biological catalysts which enhance the rate of biochemical reaction, and that is the reaction taking place in the organisms. These are generally proteins and even under the thermodynamically unfavorable conditions. For example, reactions which generally occur under the drastic conditions occur under normal conditions of temperature and pressure in animal and plant bodies in their presence. Enzymes participate in biochemical reactions without destruction or irreversible modifications during reaction, and that is why they are considered biological catalysts. Word enzyme is derived from the Greek word and which stands for yeast and was coined by Kohne in 1878 to define the fermentation of yeast cell. Enzymes differ from the catalyst in the sense that they catalyze biological process and undergo change but are ultimately set free. According to Eyring's transition state theory of reaction proceeds to the high energy transition states, and transition state is reached through the kinetic energy of the reactants, and the energy is called activation. The required energy is called activation energy, and which may be determined from the plot of logarithm of reaction rate versus temperature. And this transition state is of maximum energy and not any intermediate compound. The concentration of activated complex at transition state determines the rate of chemical reaction, and the role of enzyme is that they reduce the energy of activation. The role of enzyme is what? It reduces the energy of activation and thereby allowing the reaction to occur at lower temperature on account of their reversible binding with reactants, and the steps involved in the enzyme catalysis of a single reaction can be represented as given below. Enzyme plus substrate form the enzyme substrate complex, and this enzyme substrate complex, this is the activated complex at transition state. Then it forms the reversibly enzyme substrate complex, and this enzyme substrate complex again is in equilibrium with the activated complex at transition state. Then enzyme product complexes, and these enzyme product complex again activated complex at transition state, and this within equilibrium forms the product and enzymes. So in enzyme catalyzed reactions, though activation energy is lowered, but net energetic and reaction remain unchanged. So here we end our lecture of the biological and chemical catalysis. Thank you.